further seeing.
Okay.
Yeah.
Okay.
Hi, Elena. I'm the Dr. Wang from Taiwan. I have a different viewpoint to the other two experts because in Taiwan, cetuximab is reimbursed in certain third-line setting, so always use irinotecan in the rechange. I think the rechange with irinotecan because if the patient is ROS1 mutation type, they will have the bevacizumab in the first-line setting, and we have a cetuximab in the third line for the reimbursement. But in Taiwan, we always use irinotecan for the third line for the rechange. So some more patients will have the response. So I don't think that the rechange irinotecan is a big issue in your study. So maybe you can just go ahead.
Mm-hmm. Also, I have another question regarding because a US physician, for example, was proposing us also to have colorectal berzosertib irinotecan in second line setting, not for all patients, of course, but this was also another option. So having a third line where there is a kind of allowed rechallenge, or have a second line where there is not, or have or privilege the let's say the gastric indication rather where you have less irinotecan pretreated patients.
Yeah, Elena, that's thank you for sharing this data. It's really interesting, and it's very interesting to explore this type of drugs because it seems to be clear that colorectal cancer needs chemo and needs targets focused on the cell cycle. So I endorse the comments raised by both Dr. Takayuki and Dr. Reichelman, and I have a question about functional models. Have you tested these combinations in functional models, either pretreated with irinotecan or not, and functional models with ATM loss or p53 loss? It's really interesting also this approach, because the design is really brave. I agree that I'm not sure that for example, if we consider that this is very difficult to find patients that have not been treated with irinotecan, that has been already commented. Indeed, I'm not sure about the comparator in this setting, and in the second line setting, it would be also difficult to find a comparator. Sometimes it's interesting to build a clinical trial with an enrichment of biomarkers if you have that data and you have assessed that in functional models, and try to build more evidence in that sense. And sometimes the design of the trials allow to include patients in earlier lines and in different cohorts. I mean, patients treated with previously treated with irinotecan or not, or that's it. But more evidence will be required.
Yeah, for sure. There was some more expectation from the company side in terms of biomarker activity, but what was seen so far, even in the clinic, in the clinical experience, there is no biomarker that has for sure a correlation. And among different studies from phase 1 to phase 2, many studies that we have had with this drug, monotherapy or combination therapy, there is no clear relation. So, and also because these mutations are not frequent, so we're talking about very small numbers. We're not yet able to really plan a sure preselection. So what is here, the intention is to have the most probable mutation to be preselected and see, because if we leave the common population, we will never see any correlation. And about the preclinical models, there was something done, but maybe not as focused on the colorectal modeling. I'm quite sure we don't have the rare challenge experiments done. Maybe this is